Hello, welcome all to the ZOOMinars on molecular basis of proteinopathies. Today's speaker is Dr. Markus Zweckstetter from the Max Planck Institute for Biophysical Chemistry, Göttingen, Germany. Markus received a distinction in physics from the Ludwig Maximilian University in Munich. Then he did his PhD at the Max Planck Institute for Biochemistry in Martinsried, Germany, and did postdoc after that at the Max Planck Institute for Biochemistry, and followed by that at NIH. Then he was awarded a DFG Emmy Noether Research Group to start his independent career at the Max Planck Institute for Biophysical Chemistry in Göttingen in 2001. In 2012, he was appointed as W3 professor at the University Medical School Göttingen and started a research group on structural biology in dementia at the German Center for Neurodegenerative Diseases. Markus has already received two major ERC grants in 2011 and 2018. His group is doing extremely exciting research very well, and his research focuses on NMR spectroscopy, structural biology, neurodegenerative diseases. Of particular interest are intrinsically disordered proteins, liquid-liquid phase separation, and membrane proteins. So with that, I would like to welcome Dr. Zweckstetter to give his ZOOMinar. Markus.
Thank you very much for a very kind introduction, Rams, and for the invitation. It's great to present our work. So I share the screen. Let's see. So I chose today to speak about our work on the protein tau and what we learned over the last couple of years using mainly NMR spectroscopy. So, of course, this story of tau goes back many years. Actually, goes back to 1901 when Auguste Deter, which is the lady here on the right-hand side, and she was actually born in a city which is very close to Göttingen, so where I'm now, so about 50 kilometers from there. So she was sent by her husband to Frankfurt to see Alois Alzheimer, which was at that time a psychiatrist there. And when Auguste then died in 1906, Alois Alzheimer got the brains of Auguste Deter and did pathology on it. And at that time, actually 1906, then he observed these, what you see on the lower right-hand side, these aggregates or deposits, which we nowadays know are neurofibrillar tangles made up of the protein tau. Another very important finding was then by Mark Kirschner's lab in 1975, when he showed that protein, which we now call tau, and which he actually designated as tau already here in this article, actually co-purifies with microtubuli. And yeah, later on then it was shown that actually this tau microtubuli associated protein is the one which makes these neurofibrillar tangles in the brain of Alzheimer patients and patients with other tauopathies. Since then, 1975, of course, there's been a lot of work ongoing, mostly using biochemistry to understand both the physiological function of tau, but also its pathological role in Alzheimer's disease and other neurodegenerative diseases. And maybe this is a cartoon summarizing some of the results. As shown on the other slide and shown by Mark Kirschner, so tau binds to microtubuli. It regulates the dynamic instability of microtubuli, and it also can nucleate actually microtubuli from non-centrosomal sites, which we'll come back later to. During disease, it gets post-translation modified, in particular hyperphosphorylated, acetylated, where it then sort of falls off, so to speak, from the microtubuli. So this interaction is perturbed. And then in solution, it can form aggregate, self-associate, and form these amyloid fibrils, and which we then can observe by pathology as neurofibrillar tangles. And of course, there are many other players involved here, which are illustrated, such as kinases, GSK3 beta, but also many other binding partners. Now, despite this now, so from 75 to now, so the last 45 years, really a lot of work has been done on tau, there's still some very basic and critical questions which we try to address currently. And this is, of course, if and how we can detect the initial phases of Alzheimer's disease, which role is played by genetic and environmental factors, because Alzheimer's disease is mostly a sporadic disease, and mutations only contribute to a small number of patients. And what I will focus today on, what are the molecular mechanisms behind tau neuronal toxicity, and can we further narrow them down? Now, why, you might ask, has it taken so long? So the protein was detected in 75. The plaques, or the tangles and the plaques, were described actually in 1906 in a meeting by Alzheimer in Tübingen. So why is it taking so long to really understand how tau causes toxicity and how we can cure the disease? And I argue here that one of the challenges in understanding tau and what it does is it's what is called here the intrinsic disorder. So shortly after tau was purified by Mark Kirschner's lab, in particular the Mandelkopf lab, spent a lot of time studying this and actually showed that in solution the tau protein is intrinsically what we now call intrinsic disorder. And the term intrinsic disorder was actually coined at that time for the tau protein. And now in 2020, we of course know that there are many intrinsically disordered proteins which play a huge role in biology and pathology. So what does this mean? Intrinsic disorder, it means that under physiological conditions, there's no rigid structure, there's no tightly packed core, and it's extremely flexible and can adapt to its environment. So I think an important step was we made actually in 2009 then, when we used NMR spectroscopy to analyze the full length 441 residue tau, which is 2N4R tau, by NMR spectroscopy and characterize its dynamic structure in solution. And NMR spectroscopy, I argue here, I think is extremely important for this aim because of this intrinsic disorder and very high flexibility of tau protein. I also want to point out the very important work by the groups from Guy Lippens and David Eliza, also studying tau by NMR spectroscopy. Now, what you see here in the background, actually, what was already on the first slide, already tells you a very important activity of the tau protein in the cell. And this is it binds microtubuli, as you can see here. So when you, for example, overexpress or bring tau into the cell and fluorescently label it, then it very nicely actually labels the microtubuli. So, of course, this has then initiated a wide range of studies trying to understand the molecular mechanisms of how tau binds to microtubuli. And for that, structural biology was used extensively. Along the many years where the tau microtubule interaction has been studied, a number of different binding modes have been revealed or proposed. For example, in a study from 2003, it was suggested that tau actually binds to the Taxol binding site on microtubuli. Electron microscopy in the old days at kind of low resolution showed that tau can sort of line along microtubuli filament. So tau would be in red, the microtubuli filament in blue, and this is the side view. And then more recently, with the sort of cryo-EM revolution, a first structural model of one repeat or two repeats of the tau protein when bound to microtubuli was revealed. Now, how does NMR see the binding of tau to microtubuli? And what I show here is basically on the x-axis, you see each individual residue, and I told you we're working with a 2N4R tau. And on the y-axis, you see an intensity ratio of signals in an NMR spectrum of tau relative to the one when you add microtubuli. So I bound would be the intensity of the NMR cross peaks in the presence of microtubuli, I free in the absence. And of course, if there's no interaction of a certain domain or even residue of tau with microtubuli, you would expect a value of 1. So which indicates that under these conditions, for example, the first 45 residues of tau at best very weakly interact or don't interact with the microtubuli lattice. In contrast, then when you follow this sort of landscape here, then when you approach the prolineage region, which are denoted here as P1 and P2, as well as the 4 or even 5 microtubuli binding repeats, then you see that there's a strong interaction. However, it's not a uniform interaction across the prolineage region and the microtubuli binding region, but you have highly localized spots of binding of tau to microtubule, and we labeled them these hotspots of the microtubule interaction as A, B, C, D, E, F. And in between these hotspots of interaction, you have regions which remain actually flexible when the tau protein is bound to microtubule. So again, this is a dynamic interaction which is sort of intimately related to the intrinsically disordered nature of the protein. So the mechanism of how tau binds to microtubule is, I think, better represented by this dynamic picture where you have these individual hotspots of tau in the tau sequence. So this would be P1, P2, and there we would have the 4 binding sites in the repeat region. And they sort of dynamically and in an interchangeable manner bind to the interface between alpha-beta tubulin heterodimers. And then, of course, when we can freeze out such a conformation by, for example, cryo-EM, then one can visualize this single bound conformation. But please keep in mind that this is a highly dynamic process in agreement with the fact that if you do microscopy on cells, that tau actually rapidly diffuses on the microtubule surface, or can rapidly diffuse. Another area where this dynamic nature of tau is extremely important is for the interaction of tau with filamentous actin. So in a physiological context, tau is mostly localized in the axon, where it associates with the microtubule lattice or with microtubule. But during disease, a number of studies have shown that tau is mislocalized to dendrites, as indicated in this cartoon here. And when mislocalized in a diseased state to dendrites, it can actually bind to filamentous actin. And this is illustrated here in an in vitro setting. So when we have G-actin and we add tau, we promote F-actin formation as well as F-actin bundling, as is indicated here. So then we used NMR spectroscopy to understand sort of how tau binds to filamentous actin in a diseased associated context. So we used NMR spectroscopy, as I mentioned, and so again, what you see here is only a short part of tau, because a large part of tau actually remains highly dynamic and complex also with F-actin. However, now you have short regions in the tau protein, actually in the repeat region of tau, which fold into alpha helices as illustrated here. And these are conformations which were derived from NMR spectroscopic measurements. And I just want to point out that the illustration shows not one actin filament, but actually 2. So this would be part of one actin filament, that's another one. So actually what tau does is it uses these local alpha helices to cross-link 2 actin filaments. So this would be one helix binding to one actin filament, and there's another one. So if you put this together for microtubule and F-actin binding, then we can come up with a scenario where tau is on one hand able to cross-link the F-actin filaments using these alpha helical conformations, but it can also bind to microtubule, shown here on the right. And finally, or thirdly, it can actually also cross-link both F-actin with microtubule, because there's some regions which are used for both of these activities, so for the F-actin activity and for microtubule activity, but there are also distinct regions, for example, here in the P2 region, which are unique to microtubule binding and are not required for F-actin interaction. So again, a very heterogeneous dynamic situation which we have here. A third system which I want to use to illustrate this dynamic nature of tau and why it has been difficult for many years really to understand on a structural level how these interactions of tau with the cytoskeleton or with chaperones occur is illustrated here. So again, using an MR spectroscopy, we studied here the interaction of tau with the chaperone system, or a binary chaperone system comprising HSP90 as basically the base, and the co-chaperone FKB51. And so the FKB51, it would be actually the one here shown here in blue, and the HSP90 in gray. And tau is illustrated here as this spaghetti kind of representation, where you have the proline region in yellow and the microtubule binding repeats in red. And what the NMR study showed is that in the complex, the microtubule the proline region shown here in yellow clusters around the PPase catalytic pocket. So in that way, the FKB51, in particular the FK1 domain, can promote this trans isomerization of tau in this HSP90 FKB51 complex. So I hope these examples really showed you and made you sort of alert to the fact that we're working here with a very dynamic system. And so what we generally get with structural biology are snapshots of these sort of very dynamic transient interactions. A final example I just want to stress is related to tau amyloid formation and tau strains. Of course, it's a highly important topic, and fantastic work from Sarah Scheres and Michel Goddard have revealed in the last 3 years, basically, a number of cryo-EM structures of amyloid fibrils extracted from deceased patient brain. And this was actually the work here, the first one by Anthony Fitzpatrick in 2017, and from an Alzheimer's patient. And what is illustrated here in blue is actually the core of fibrils, right? But if you look at the residue number, this goes from 306 to 378. So we're talking about 80 residues for a 441 residue protein. And that there's maybe more to it than just this very rigid core is also illustrated here by this additional electron density, which was detected but couldn't be identified. And probably even more important from a pathological point of view, there are monoclonal antibodies developed by Peter Davis' lab, such as ALZ50 and MC1, which bind pathological tau in patient tissue. And these two monoclonal antibodies, they require a discontinuous epitope, which consists on one hand of these residues here, which is actually, as you can see, part of this cryo-EM structure, right? But it also is absolutely dependent on a second epitope here, which is here at the N-terminus and involves about residue around residue 20. So one needs not only this, but actually also this epitope to really be able to detect it by these monoclonal antibodies, which indicates really there's a large part of tau which is not seen in the cryo-EM structure, but which could, of course, be important for neural toxicity and also spreading. So while this was more a historical view, I would say, so far, I now want to discuss a little bit our thoughts about the potential role of tau condensation in disease. So what is that for those who haven't come into contact with this? So if you look back into biochemistry books, and maybe which are still published nowadays, of course, there's a very nice descriptions of membrane-bound organelles such as mitochondria, ER, and Golgi. But in recent years, it has become clear that besides these membrane-bound organelles, there's a large number of compartments in the cell which are not surrounded by membranes. And these include nucleoli, P granules, stress granules, and several other ones. And a large number of very nice studies have shown in recent years that these membraneless organelles or biomolecular condensates, they are formed or driven by the a process which is called liquid-liquid phase separation. And this liquid-liquid phase separation is really strongly dependent on intrinsically disordered proteins. So actually in 2017, then we showed that the microtubule binding repeats of tau can undergo phase separation. And this is illustrated here in this little movie. I can go back, as you see, right? So you have this, if you look here, you have droplets and they fuse, right? They are round. So they are typically of what one would expect for example, if you mix oil and vinegar. In parallel, both or a number of other groups, such as from Ken Kosick, Song Yi Han, and from Brett Hyman, have also shown that tau can undergo phase separation. So more recently, we were particularly interested in RNA-mediated tau condensation, and this just illustrates that different parts of tau, so is it now just the microtubule binding domain or just the projection domain, which we call the construct K25, or the full-length protein, from very nice, phase-separated compartments in the presence of RNA. You can alternatively, instead of RNA, you can also use dextran. And tau, because of its sort of unique charge distribution, that the N-terminus is negatively charged and the repeat region is mostly positively charged. Actually, tau also phase separates at low ionic strengths, also in buffer, without addition of dextran or poly-U. Nevertheless, so one can get phase separation for these different constructs using RNA. However, these tau condensates are not all the same in terms of the biophysical properties. And this is illustrated here. So we did fluorescent recovery after photobleaching here for both RNA-induced droplets of K18, so the repeat region, as well as the full-length tau protein. And as you can see, if you look at the fluorescent recovery curve, then the fluorescence for K18, this is also illustrated here, recovers fully, right? Whereas for tau, h-tau40, on this time scale, the recovery is slower. And so where does this come from? We believe that this is related to the unique differences in amino acid composition. So whereas for a K18, you have only about 1% of arginines and more lysines, this sort of balance between lysines and arginines is sort of tweaked towards arginines, which are known to have a stronger interaction with RNA when compared to lysine residues. So is this of any relevance, these in vitro experiments for the cell? As I pointed out, one very important membrane-less compartment in the cell are stress granules. And Ben Wolosin's group and others have shown that tau co-localizes with stress granules in patient tissue. And this might also be, this interplay between tau and stress granules might be important for the pathogenic processes in Alzheimer's. So what we just show here, and these are actually experiments done by Saskia Hutton in Dorothy Dorman's lab. We use the semi-permeabilized cell assay, and we mark the stress granules in stressed cells here with the stress granule specific protein G3BP1 with an antibody. And when you stress the cells, you see you get nice stress granules. And then you can also visualize the fluorescently labeled tau, and you do the merging. And then you see that in agreement with the data from Ben Wolosin, tau actually can nicely co-localize with stress granules. However, we use the semi-permeable cell assay to understand a little bit more which domains of tau are particularly important for this. And there we found that the colocalization amount is similar for just the repeat region as well as for full-length tau, but it's actually increased when we just use the projection domain. So if one takes all these three together, right, because the full-length tau also contains the projection domain, it actually suggests that the repeat region sort of attenuates the colocalization of tau with stress granules, which could, for example, be because the repeat region is very important for binding to microtubuli, and such there's competition between microtubuli binding and stress granule binding. So when we talk about tau and neurodegeneration, of course, it's aggregation into insoluble deposits, the neurofibril tangles, is of prime importance. So we then asked if it's actually possible, if the tau can convert from this liquid-liquid phase separated state, from these droplets, into full-blown amyloid fibrils. And for that, we first prepared, again, our droplets here. It's shown here on the top, actually on the right. And then we added to the slide heparin, which is a known aggregation inhibitor, enhancer of tau. And as you can see, if you just compare these two pictures, it's actually the same slide, just two different parts. So on the left, we added the heparin. On the right, there's no heparin. Then you see that right after addition of heparin, the morphology of the phase separated state changes. And this change in the morphology continues after 30 minutes, after 24 hours, and after 3 days. Actually, we have full-blown amyloid fibrils. So I think the process or these images here are in line with sort of a conversion from a highly dynamic fluid state, as is shown before by the FRAB, into a solid-like phase. So that is sort of maybe nice pictures, but it's, on the other hand, it's just pictures. So in order to further support the connection between condensation and amyloid formation, we sort of went back to now the conditions which we used actually for phase separation, and thought to establish a correlation between the conditions for liquid-liquid phase separation, LLPS, and amyloid fibril formation. So we changed on one hand the temperature, which is here the left column, or the ionic strength, the right column, and also the pH data, and the pH which are not shown here. And in phase separated liquid conditions, so in the droplets, the THT fluorescent is low, indicating that there are no amyloids yet formed, right? So this is a dynamic state in which the tau is still largely intrinsically disordered. However, there's a clear dependence of LLPS formation on both the temperature and the ionic strength. And I just want to point out or draw your attention here to the temperature, where if you are below 30 degrees, even with heparin, right? So here we now add heparin in the optimal ratio of 4:1. So even when you're at 30 degrees, right, you are with heparin in the optimal ratio, there's almost no LLPS, and there's very little amyloid formation. Then you get a maximum in both LLPS and amyloid formation at around 37 degrees. But when you further raise the temperature, then again you lose both the liquid phase separation as well as the amyloid formation, which to us at least suggested that there is a connection between these two processes. So when we talk about tau, and I just show you here in the bottom again, you immediately notice once you look at the amino acid sequence that it really contains a lot of lysines, which are indicated here by these arrows, right? And when we then further think about post-translational modification, acetylation is probably the first modification which we think about. And a number of years ago, I think in 2011 now, the group of Li Gan has shown that acetylation is connected to the neurodegenerative process of tau. So it inhibits degradation, it contributes to tauopathy, and to cognitive defects. So we therefore ask, does tau acetylation affect tau phase separation? And yes, it does. We first basically mix tau with RNA, which would be the green and the blue circles or dots. Then they phase separate, as I showed you before. And then we added the enzyme acetyl-CoA, which acetylates tau-lysine residues. And then what we observe is here shown in a cartoon and here below by microscopy, actually the droplets dissolve. Right. Similarly, you can also acetylate tau first, and then try phase separation again. It interferes with the acetylation interferes with tau phase separation in solution. And this was seen both by us and by Ferion. So why does tau weaken this RNA-mediated, why does acetylation weaken this RNA-mediated tau phase separation? For that, we resorted to NMR spectroscopy. So we probed the interaction of tRNA with full-length tau, as indicated here, for two residues, alanine 119 and lysine 317. And then we did the same analysis for wild-type tau or unmodified tau and acetylated tau. And the results is shown here. In black, you see the NMR binding profile of the unmodified tau with tRNA. And first of all, what to recognize, the binding is exclusively related to the residues to the range from residue 120 to about 400, which makes perfect sense because that's the part of tau which contains most of the lysine and arginine residues and has at pH 7 a positive net charge. I just also want to point out that a particular strong interaction with RNA you find for the proline-rich domain of tau. And then when you do the same analysis for acetylated tau, the interaction is weakened, as indicated here by this decrease in this histogram plots. Now you might wonder, okay, but there's still binding, of course, so it's not completely lost. This is also expected because we still have the arginine residues, which are indicated here by these downward-looking areas, which of course are not modified by acetylation and do contribute to RNA binding. This was a pure in vitro experiment. So then we now ask next if acetylation affects the association of tau with stress granules. So we again asked Saskia Hutton and Dorothy Dorman, and they performed the semi-permeabilized cell assay where you have your cells, you digitize them with, you permeabilize them with digitonin, and then you close sort of the nuclear pore by WGA, and then you add your protein of interest, which in our case was tau, either unmodified or acetylated. And then you can see here that we again have our stress granules, which label for both G3BP1 and TR1. And H-tau40 is also, the unmodified is present in the stress granules, whereas the acetylated tau, there's basically no colocalization with the stress granule at all. And we can quantify this, and you see the labeling for acetylated tau is similar to the background, which shows that both in vitro and in the cells, the acetylation really blocks the association or the phase separation of tau and the association with membraneless compartments. And now, what does that mean for neurodegeneration? That's a very interesting question, and I think interesting to discuss. Of course, on the slide before, I showed you that acetylation enhances, let's say, neurotoxicity of tau. And here we show that acetylation decreases association with stress granules. So one hypothesis could be that actually the stress granules in Alzheimer's disease or in these patients are not exclusively pathogenic in a sense, or have a pathogenic role, but might actually have a protective role also, which then soak away the tau, and such tau cannot do any harm to the cell. But of course, this requires further experiments. Now, I started with acetylation. You might have wondered why, because of course, the best characterized post-translational modification of tau is phosphorylation. And there are a number of monoclonal antibodies, such as 88, 100, 180, which recognize epitopes in different parts of tau, and you can use them for visualizing deposits of tau or pathologic tau in patient tissue. So we, of course, asked how does phosphorylation affect phase separation of tau. We, in this specific case, used the repeat domain of tau and phosphorylated the repeat domain by the kinase MARC2, and the dots here indicates the site of phosphorylation. And then we repeated our in vitro phase separation experiments with this MARC2 phosphorylated K18. And we again got very nice droplets, which formed in a time-dependent way. And you also see oswald ripening, so they grow. But what you don't now see, because I don't show you the reference here for if we do the phase separation with unmodified K18 at 2 micromolar, we don't see droplets. So we need higher concentrations, whereas with a phosphorylated MARC2 phosphorylated K18, we can go down to 2 micromolar and still are able to get phase separated state. So the MARC2 phosphorylation in the repeat region strongly promotes the phase separation of the repeat domain of tau. Another experiment which we did, I don't want in detail, but just to point out that we can make droplets with phosphorylated tau, and this then recruits unphosphorylated tau. So even if there's a small part of tau phosphorylated, this would overall enhance the phase separating capability of tau. So while we were working on this in 2017, there was also a study done in Anthony Hyman's lab, and they also showed that tau phase separates, and they were particularly interested in the ability of tau to nucleate microtubule non-centrosomal sites. So what they showed is illustrated here in a cartoon. So tau phase separates, as you've seen before, and then when you add tubulin heterodimers together with GTP, they concentrate within the tau droplets. And then what you see is actually better illustrated here than in the cartoon, right? You see that these droplets deform, as indicated here by the red arrows, and ultimately you get a very nice looking microtubule filaments. So we repeated this experiment with our protein, and this is the result here using both phase contrast microscopy and fluorescent microscopy. We start with droplets, as you can see here, of tau. The tubulin strongly concentrates into the tau droplets. And then after a few minutes, we get very efficient microtubule formation. And after 30 millimeters, actually, we already have an extremely beautiful looking microtubule. And tau is actually binding to this microtubule, as you can see here. So then we ask, how do post-translation modification affect this extremely efficient microtubule assembly from tau phase separated compartments? And so again, going back to these different types of phosphorylation here, we actually picked one, and this is related to phosphorylation here in the prolineage region. In particular, we were interested in the phosphorylation which is recognized by the AT188 epitope, and which requires definitely a phosphorylation 3 in 231 and maybe a contribution of phosphorylation serine 235. And we picked this particular phosphorylation from the many which you see here because actually Gillippin's group showed in 2009 that this phosphorylation here by CDK2 can strongly attenuate microtubule formation in the absence of phase separation. So we now wondered, okay, in the phase separated conditions, right, you increase the concentration of tau 10 times, you increase the concentration of tubulin 10 times. So shouldn't this overcome this barrier which was shown here? So we phosphorylated tau by CDK2 and identified the sites of phosphorylation here by NMR spectroscopy in a quantitative manner. And then investigated if the CDK2 phosphorylated tau can undergo phase separation. And it truly does. Actually, it again has a higher propensity for phase separation, as I showed you before, when compared to the unmodified protein. But when we then do the microtubule polymerization assay, so this LLPS-based microtubule polymerization assay, again, we get nice droplets, tubulin concentrates in the these tau droplets of phospho-tau, right? But we can wait, and there's absolutely no microtubule formation, right? So despite the fact that you have really a very strong increase of both tau and tubulin in the droplets, it is not anymore able to polymerize or assemble into microtubule. We then asked if it is specific to the phosphorylation at 3 in 231 and serine 235. So when we made a mutation, we actually introduced it in the 3-repeat isoform tau HR23. We first checked that HR23 also polymerizes or can polymerize tubulin into microtubule, and this is indicated here. And then we introduced the mutation, and in line, consistent with the CDK2 phosphorylation, it fully blocks the polymerization of tubulin into microtubule. So why is this, we then asked, and for that we used again NMR spectroscopy. So what you again see here, you're already familiar with it. You see the NMR binding profile of tau shown here in black in the presence of tubulin now, unpolymerized tubulin. And in pink, you see the broadening profile for the CDK2 phosphorylated tau. And as you can see, so in the beginning, it's the same. Also here in R3, R4, R prime, the broadening profile is the same. But in particular, in P2 and R1, there's a strong difference in the amount of NMR line broadening between the unmodified and the CDK2 phosphorylated tau. it shows really that this is specific to the P2 region, sorry. Just to show that this is really specific, we also used different kinases. So I showed you before the MARC kinase, which phosphorylates the repeat region here. We quantified this. It does not block this LLPS-mediated tubulin assembly. And also importantly, tyrosine phosphorylation by c-Abel here at these 4 tyrosines in tau also has no effect on microtubule polymerization from tau condensates. So this allowed us to arrive at the following conclusions. So active tau is able to phase separate into these droplets where the tau concentration is increased strongly. When one adds tubulin, or when tubulin comes close to this, it concentrates into these phase separated compartments together with GFDP, and it can form or the tubulin assembles into microtubule. In contrast, when we have disease-associated AT180 phosphorylation, you still get phase separation. Actually, it's promoted. Tubulin still concentrates in, but it's absolutely impossible anymore to polymerize into microtubule. And actually, with some further NMR experiments, we could show that this is related to salt bridge formation, intramolecular salt bridge formation between the phosphorylated threonine residue 231 and nearby arginine residues. So if I put all of this together, then this would be the findings, or how we see this currently is you have monomeric tau. It's extremely soluble, right? Which is very important to remember, right? I mean, you can keep tau on the bench for a month without having any sign of aggregation, which is of course very surprising given that you find it in the brain of patients with Alzheimer's disease aggregated. Alternative splicing can influence tau. So 4-repeat tau has a higher propensity for phase separation than 3-repeat tau. We and others showed that phosphorylation at different sites actually promotes tau phase separation. On the other hand, acetylation really blocks the ability of tau to undergo phase separation. Once the droplets are formed, tubulin concentrates in there and efficiently polymerizes microtubule. And in addition, heparin can go in there, and in this high protein dense, high concentration protein dense droplet, heparin is concentrated, and then tau tangles can form. So I want to end with sort of, again, a historical view a little bit. Also, I didn't really show many NMR spectra during this talk. Actually, I mean, there's a large part of the results I presented were based on NMR spectroscopy. Now, if you look back in 1951, 70 years, so to speak, there's a picture of an NMR spectrometer shown here, which was operated at a proton frequency of 30 megahertz, right? So I think we not only made a big step forward in understanding of tau on a structural level, but also in NMR spectroscopy during these 70 years. So actually in July this year, we received a 1.2 gigahertz NMR spectrometer in Göttingen. So this is Christian Griesinger, by the way, and it's myself. In about 70 years or so, the magnetic field strength, which is now available for biomolecular NMR spectroscopy, increased 40-fold. Right. So what does this give you? This is a little bit shown here. So that's now a picture from this week, actually, taken by Lauren Andreas. And actually, unfortunately, I have to say that the magnet quenched in the meantime, but this spectra were recorded before the magnet quenched. And it's a comparison of a proton-nitrogen correlation of one of the membrane proteins Lauren works on, using proton detection, so high-fast MIS, 100 kilohertz spinning, either recorded at 950 megahertz or at 1.2 gigahertz, shown in red. And I think what is very nicely seen is that you really get significantly sharper lines. So for me as a solution state NMR spectroscopist, I mean, this almost looks like solution state NMR. And we still have to quantify it exactly, but also the sensitivity seems to be at least improved according to the magnetic field strength. So we're very much looking forward to get the magnet again up to field and do further measurements. So with that, I'm at the end, but not before. I really want to thank all people in my group. As you see here, a more or less recent group picture. Because, of course, I mean, I showed a number of studies in the result, or at least the results of these studies. But at the end of the day, it's really, I think, a combined effort of the whole lab where we try to understand proteinopathies using a mass spectroscopy. I also want to particularly thank Eckhard Mandelkopf, which we collaborated over many years, as well as the funding agencies, in particular the European Research Council. Without the funding from the ERC, this work would not have been possible. And I thank you very much for your attention, and I'm happy to take your questions.
Markus, thank you very much for a great talk and sharing your exciting findings, as well as the new magnet and new data as well. That's exciting. Okay, for Q&A, for participants, there are two ways to ask questions. One is to directly raise your hand and join the panel and talk to the speaker directly, or you can post your questions in the Q&A folder. We can ask the question on your behalf. I strongly encourage students and postdocs to participate. You will be given the first priority. If you raise your hand, you can join the panel, or also you can join us after 11:10 or so as well. Okay, let's see. Well, there are several questions. Let's go with the ones in the attendees. Samrat, you can ask the question directly. Samrat, can you hear us?
Yes.
Yeah, go ahead.
Hi, Markus. This is
Hi, Samrat.
Hi, Markus. This is a brilliant talk. So I really enjoyed listening to your terrific talk. So the question is, when you add heparin to the phase-separated droplets, you slowly see the transition to amyloid fibrils. Now, the question is how these amyloid fibrils are related to the de novo amyloid fibrils without phase separation. So is there a structural distinction between phase-separated amyloids and de novo amyloids?
Yeah, very important question. And this is an ongoing project which we're looking at, but from what we see, actually, the structure seems to be different. So we're very excited to that. I mean, I cannot really tell now much more, but of course, that's a very important question. I mean, showing that you have different tau amyloid conformation in different diseases. Yeah, and a critical question is really where I think many people work on what causes different tau amyloid structures in different patients.
Okay, thanks. So it's like, oh, sorry. I mean,
go ahead.
So sorry. So seems like the very transient interactions that are present in liquid-like droplets, they are now converting into more persistent interactions in the amyloid state. So do you think the electrostatics become quite predominant in the amyloid state as opposed to the phase-separated liquid-like state?
I mean, currently I don't have really very strong data for this, but from what we see so far, using any mass spectroscopy in the phase-separated state, I would be in favor more of hydrophobic interactions strengthening, right?
Okay.
Okay. So there's some NMR data which point to that. We see changes, but we haven't really fully understood where these changes in structure and dynamic come from. Yeah.
Okay. Thanks, Markus.
Thanks, Samrat.
Next, Nalini, can you ask the question directly?
Sure, sir. Thank you for this wonderful talk. So I have a small question regarding the consequences behind the stress granule formation. So most of the study shows the role of molecular crowding as well in the phase separation process. But in vivo condition, there is a relative abundance of tau during Alzheimer's disease that drives its aggregates formation rather than going towards the stress granule formation. So in that case, what would be the consequences of events that lead to the formation of stress granules by tau rather than directing its aggregates formation?
Maybe I was not very clear about that. I think I think what I didn't want to claim, and I think what was also not supported by the data strongly, at least, is that tau itself causes stress granule formation, right? I mean, stress granule formation is caused by many factors, and there are other proteins such as G3PP1 and TR1, which are extremely important for stress granule formation. Tau is more associating with the stress granules, right? And so, and that's, I think, I think the final verdict is still out if this association of tau with stress granules is beneficial or detrimental for the cell.
Yes, sir, because some of the studies says that stress granule formation is good, but in case of neurodegenerative diseases, we mostly hear that it's detrimental for the cells. So
yeah, so that exactly. So in other, in let's say ALS related research, of course, the data has been shown that if stress granules, let's say, age in that sense and are not reversible anymore, that this can be toxic for the cell, right?
yes.
But I mean, to me, I think there's definitely more studies warranted which investigate whether these stress granules and maybe different types of stress granules, right? There are different types of stress granules in the cell where they are contribute really to the toxicity in a positive or a negative sense. Yeah.
Yeah, sure. Okay. So maybe it could be the composition of the stress granule at a given particular pathological condition.
For example, yeah.
Okay, thanks
sure,
for the question. So, Christian, can you unmute yourself and ask a question? Okay, it looks like, okay, Sudipta, go ahead. Sudipta Maiti, go ahead and ask question directly, yeah.
Hi, Markus. Good seeing you again for a long time.
Good to see you.
Okay, so fantastic talk. I was just interested in that a little bit, where you said that the phosphorylated tau, which forms from the LLPS, recruits unphosphorylated tau into it. Mm-hmm. If I think of from a thermodynamic standpoint, is that because there is kind of a nucleation barrier that the phosphorylated tau helps overcome? Because ultimately it's phase transition, and then if it is above its saturation concentration, then unphosphorylated tau shouldn't get into it. But it could be that unphosphorylated tau is always in a metastable state, and it's the nucleation by phosphorylated that helps it come in. How do you think about that?
That's an interesting question. I mean, we haven't investigated this. So what of course could be, or what could contribute, is that, I mean, that the phosphorylated tau has let's say an increased binding property to the unmodified one, right? Because of course electrostatics plays an important role. The repeat region is particularly, but this was Mark. No, this was both Mark phosphorylated one. Yeah, very interesting question. I don't have a good answer now, I have to say.
Okay, I'll chat you through later.
Sorry, very good question. Yeah, thank you.
Thank you.
Okay, Binzu.
Hi, can you hear me?
Yeah.
Hi, great talk. I have a question about acetylation. So you mentioned in your stress granule experiment showing acetylation of tau blocks association with stress granule. And as most of people in the field think, stress granule mostly, leads to neurodegeneration. So it seems as if that's like opposite direction. Maybe not necessary, as some people will say, stress granule sometimes will be protective instead of toxic. So what's your your construct of the human tau, acetylated? Is it acetylated ubiquitously? Because there are dozens of acetylation sites. From one of the recent study by Fitzpatrick from Columbia, from their cryo-EM study, cryo-EM and mass spec study showing there are limited sites acetylated. Number of them are showing in repeat region. So I was wondering whether your construct, the acetylation, whether it's site specific, can contribute to that, to how to explain of that association with stress granule.
Yeah, so I think there are two potential approaches to this. First of all, what you mentioned, right? I mean, there is a suggestion and also some data which point to a, let's say, pathogenic contribution of stress granules to neurodegenerative disease, right? But to me, at least, the final verdict hasn't been made. So, if we favor or if we argue in favor of the positive role of stress granules, right, then the blockage of the binding of acylated tau or colocalization with stress granules would be consistent. If it's a sort of a toxic role of stress granules in neurodegeneration, then of course one should try to explain it. And one way would be the sites of acetylation. So what we did in particularly here is we used enzymes, so P300 or CREB, to acetylate the protein. And so this is not acetylated at the unique site, which would anyway be difficult because from at least all experiments which we did, the acetylation, at least by these two enzymes, is quite ubiquitous or not very residue specific. So of course now we could and we should try to dissect this by doing mutations and hoping that the mutations really capture the acetylation. Along these lines, right, for me this was really a little bit puzzling also because tau also has some arginines which are not modified by acetylation, and the interaction strength of arginines with RNA is stronger than with lysines. So, and the arginines are not modified. So one could have expected that the acetylation does not block, at least if the binding to the stress granules is driven by RNA, not by other factors.
Thank you.
Okay, Wang, you can ask the question. Wang, are you there?
Hi, Markus.
Yeah, go ahead.
Nice talk. Just curious, where do you expect to see this liquid phase separation in human bodies? Like how is both in neurons and secreted, and heparin will be much higher in the serum or extracellular than inside the cells.
Yeah. So,
but the concentration is kind of nanomole range, but liquid phase separation, you need micromole list.
Yeah. No, so fair question. So, I mean, we're not cell biology lab, so that's not really our strengths. But so, I mean, in the study from Susanne Wickman and Brett and Anthony Hyman, they also looked in cells. I think provided some suggestion that also the phase separation occurs in cells. There was also a recent study from Ken Kosick using the Cry2, so this blue light-induced phase separation in cells. And maybe very important also observation was, which I didn't point out in our microscopy images, but which one also sees is that you also get, let's say, condensates or blobs of tau on the microtubuli. And this was also shown, now I've forgotten the name, but by your colleagues in San Francisco, that the tau forms condensates on the microtubuli. And I mean, previously people believed these were oligomeric taus in the old studies in 2000 or 1990. But the microscopy, I think, now show clearly that these are tau condensates, which particularly occur at sort of breakage points or imperfections in the microtubule areas. And of course, there's more stuff to investigate for this, the contribution of this, for example, microtubule associated tau condensates to the disease process.
Okay, Christian Grisinger, you should be able to ask question now. I think previously I selected another question, I think.
No, it's me.
Okay, go ahead. Why did you coin the magnet?
What's that? What was the question?
Why did you coin the magnet?
Well, I mean, it just happened by chance, as it could always be. But I think my question has already been answered, namely the acetylation. So there are many acetylation sites. It's not like in alpha-synuclein, just the N-terminus, or that is acetylated in tau.
I mean, there's, as in alpha-synuclein, right, there is a sequence dependence. So in alpha-synuclein, the lysines are mostly in the first 100 residues or first 80. In tau, it's mostly in the central part. So it's also, let's say, spatially localized. And as was mentioned before, actually in Fitzpatrick's recent cryo analysis, it was combined with mass spec, actually it was found that in the repeat region, there are some lysines which are acetylated, yeah.
So it's mainly lysines, not like serines or something like that. It's just lysines.
I'm not aware of that people have shown that serines are acetylated.
I had another question because, I mean, some of the questions were also a little bit philosophical. I mean, there's normally when people look at development of patients, also they say that first A beta aggregates and that A beta somehow initiates tau aggregation. But in your talk, I didn't see then A beta in that game at all. Where would it come in?
I mean, to me, this connection makes perfect sense because, of course, you have the mutations in presenilin and so on in patients which get Alzheimer's disease. So there's no doubt that there's a very strong genetic evidence for the involvement of A beta and APPP. But I think it's not a direct interaction where you, in the sense of you mix the two proteins and they accelerate aggregation.
No, sure. Yeah, clear. But somehow.
No, but how I think, I mean, there have been a number of suggestions being made, right? So, for example, dysregulation of kinases such as MARC2 has been suggested, and there are publications which show data for this. There are, of course, other candidates such as Fin kinase in the dendrites, where somehow it could come together. But yeah, where exactly, I think people would like to really find out, yes.
Okay, thanks.
If it's not multiple hits, actually.
What?
If it's not multiple hits, right? I mean, it could be multiple pathways which then converge on tau, which I think is most likely currently a scenario.
Okay, next question, Long Cheng.
Hi, Max. This is a great talk. Thank you so much. And here I have two questions. One question is for ubiquitination, because a lot of data to point out the ubiquitination actually is not just mediate UPS, and also to modulate this intermolecular modification. Do you ever try because I know a lot of this same residue to also be ubiquitinated, also from the EM data to show that core region also is ubiquitinated. So I'm wondering, do you ever look at the acetylation or ubiquitination as a synergistic effects or have competition? And what's this, whether same role, how ubiquitination contribute this structure? And that's my first question. My second question is because here the lipid separase actually is a membrane-free system, and a lot of genomic data and to show our actual lipid metabolism. Also, a lot of tau intake, secretion, or dynamic is really organelle, like endosome involved. So, what's your thoughts? Like, what's the lipid or membrane-associated organelle contribute this dynamic process of lipid, separation? Yeah, yeah, that's my question. Thank you.
Very good questions. So we don't have really any data which I could now use for supporting my arguments. My personal opinion is, or feeling, it's not even opinion, a feeling, is that maybe ubiquitination might be quite late and mostly maybe important for degradation. Of course, there has been the ubiquitination or the of the tau fibers by cryo-EM seen or suggested, which of course could be a late-stage process. What the contribution of ubiquitination, let's say in the early phases of misfolding in aggregation and phase separation are, I'm not aware that people have looked at this in detail, but I'm happy to be corrected. In terms of lipids, yeah, I agree, and there are many because tau, right? I mean, it's highly concentrated in the cell. It's intrinsically disordered. It can bind to many binding partners. I mean, as I said, I'm a very noir spectroscopist. But if you remember this one picture which I showed you, basically as my background, I mean, if you add tau to the cells, then I mean it's binding to the microtubule. So that's definitely a good binding partner. I mean, this binding is much stronger because, for example, I might show this here, which, if I may share my screen again, just do this, do this again, because then I can actually illustrate it here. You see my screen?
Yeah.
So actually this microscope image, right? So the green one is the microtubule.
Yeah.
And the red is actually the mitochondria.
Oh, yeah.
And there's basically, we didn't really observe any strong colocalizations between tau and mitochondria. And I mean, if you compare these pictures, I think it's quite obvious. Of course, it can bind maybe to mitochondria and so on under specific conditions, and maybe also other organelles. But yeah, that's what would be my opinion. I mean,
for this imaging, did you do the extraction before you imaging, or just like a total cytoplasmic protein?
No, this is again actually the semi-permeabilized cell assay.
Okay,
where we permeabilize the cell and then
Gotcha. Thank you.
Okay, so it's close to 11:10. Let's thank the speaker for a wonderful talk. We'll definitely continue the discussion in the informal session in 30 seconds. We all would like to thank all the attendees, and please note the time change from next week onwards. We're ending the daylight saving time, so fall back so we gain 1 hour, sleep better, and stay safe. Join us the next week. Now we are switching over to the informal session. So Markus, can you look at Q&A folder? There are several questions which are not answered yet. And we go through that, and then we take more questions from the attendees as well. There's one from, we answered Wang's questions. Galina is, I think she or he wants to join the panel. I will promote her to the panel. Galina, you can ask the question directly. Hello, Galina. Unmute. Yeah, go ahead.
Thank you very much. That was a very interesting talk. So I have 3 short questions, really. So for the first one, is there any pattern or specific phosphorylation site that actually prevents the of tau? And then is there any modification that can happen after recruitment of tau into the LLDS, so after the phase separation? And for the last one, I was hoping that maybe you could clarify the recent contradicting reports about the LLPF leading to the fibrillation. That was one of the studies from last year. They identified the subset of conditions that are overlapping both for LLPF and amyloid formation of tau, but those two processes were separate.
Okay, the first question I didn't fully understand because I only heard which phosphorylation and which sites, but I didn't understand now on which effect, on aggregation or on
LLPS. So
on LLPS. Yeah. Okay. Okay.
Patterns.
For phosphorylation. We haven't identified any so far, right? So we see that Mark II phosphorylation of the repeat domain enhances phase separation, at least when we use the repeat construct. We also see that CDK2 phosphorylation, so in the prolineage region, enhances phase separation. We haven't seen a phosphorylation which, for example, decreases phase separation, but because, yeah, I mean, the c-Abl, the tyrosine phosphorylation, and so on, all of that. Sorry? Are you still there?
For example, on the series 2.
On the?
The PS262, for example, that's something that prevents aggregation. So would you think that it might actually perturb the formation of LLPS2?
Yeah, actually the 262 is phosphorylated by a mark. So there is a sort of a, let's say, disconnection between phase separation and aggregation because phosphorylation serine 262 attenuates amyloid formation, but it appears to enhance liquid-liquid phase separation. So it really depends on the site of phosphorylation, right? Because as I said, I mean, for example, CDK2 phosphorylation, it enhances phase separation. So that would be my answer to the first question. The second one I have forgotten, and the third one with the study from Song-Yi Han, I guess you referred to. I mean, on one hand, of course, this is always, let's say, on whether you look at whether the glass is half full or half empty, because even in that paper, I think there was a range of conditions which they tested where there was overlap. But on the other hand, now I don't remember now exactly which tau construct they used, because at one point they used the shorter tau construct. So that one would have to look back. So in the data which I showed, we used the full length tau construct. Yeah, and there, as I said, I mean the data are pretty clear that there's a clear overlap in the physiochemical chemistry or chemical conditions where we have phase separation, heparin-induced aggregation.
So it's not necessarily causative that LLPS would lead to amyloid formation, but it might be two processes occurring alongside each other.
No, this is, I think, I mean, these are correlations, right? So I don't want to claim a causative relationship from a correlation. Yeah.
Okay, thanks, Galina. Any questions from the panel? Markus, it looks like very rich dynamics there intrinsically in all the proteins that you're working on. Did you get to characterize the dynamics using NMR as a wonderful tool? So,
yeah, this is an ongoing project actually. So yeah, that's really a project which is currently ongoing in the lab where we try to understand the dynamics and how this changes under different conditions. Yeah.
So it must be really very fast in the NMR timescale, the fact that you were able to see the resonances using solution MR. Upon binding with, let's say, actin, it looks like
no, actually, no, I didn't go into detail there. So there we use transfer NVs.
Oh, okay.
So both for the microtubuli in order to derive the conformation of tau bound to microtubule as well as to actin, we use transfer and measurements because otherwise it would not be possible.
So what is the change in time scale of dynamics there? Is it microsecond to millisecond or nanosecond to microsecond, or do you have any idea on that?
Upon binding, you mean?
Mm-hmm.
We don't really know, right? Because we have tried, and other people I think have also tried. It's not so easy to visualize tau in the bound state using solid state NMR spectroscopy, for example. So we don't really know what the dynamics. I mean, one can do fluorescent microscopy in terms of dynamics on the microtubule bound state, but that's mostly diffusion and not, let's say, intramolecular dynamics. So we don't really know. So we know we have an idea of what's in solution, but in the bound to actin or microtubule, we don't know currently.
Okay, Magda and John, if you have any questions, I'm going to promote Bin Zhu to the panel. You can ask question directly. Bin, are you there?
Yes, I'm here.
Go ahead. You raised your hand. Go ahead.
Yeah, thank you. Hi, Magnus. I have two more small questions.
Yeah.
So the first one is about, you mentioned various segments of tau, their contribution to aggregation. And traditionally it's thought are the repeat domain, but I think your data suggesting some new roles for the P1, P2 region, some of those may contribute to. So that's very exciting. Have you tried the N1, N2, the very end of N terminus? This seems like very all disordered. Do they have any rules in terms of aggregation, like such as kinetics of aggregation? Have you tried anything about, some, for example, truncation constructs?
I mean, we ourselves haven't done so. At least not quantitative, right? The only thing I can say is if one uses a construct which comprises the proline region and the repeat domain, right? I mean, you also get nice aggregation, but I don't have any kinetic data now to really say how much different parts of, let's say, the inserts or so affect the speed of aggregation. Yeah.
Okay. Okay, yeah, I'm more thinking, most people think those are not involved in aggregation. So, but I guess,
Yeah. I think in the final state, I think that's we see, right? I mean, there we have NMR data where we actually show that really the epitope, which is recognized by ALS50 and MC1 in the N terminus, this really interacts with the core region, right? So there's really a direct interaction which provides sort of this motif which then the antibody can capture. Yeah.
The second question I have is related to the start from monomeric tau to tau aggregation, the fibril formation. So the intermediate step, most people think there is oligomer involved. And in your big diagram showing, liquid separation, the droplet formation, I was wondering whether there is any clear distinction between those lipid drops formation versus like oligomer formation, or are they overlapped?
Yeah. Yeah. Thank you for pointing this out. The oligomer issue is a complicated issue, I would say. First of all, because there is not a good, I mean, it's a very heterogeneous species, which makes it difficult to describe, right? So there's, for example, this term high molecular weight tau, and which was used in injection experiments and recently, for example, I mean, and I think we don't really know what this is, to be honest, right? I mean, it could be already partially with cross beta structure or not. So yeah, it's an important aspect. I just want to say in favor, let's say, of the phase separation is that, I mean, this really the concentration of tau in membraneless compartments, such as, for example, stress granules, right? I mean, you can potentially have a huge increase in concentration, right? And if you ever work with tau in the lab in an in vitro setting, right? I mean, in principle, if you don't have heparin, it doesn't aggregate at all over a month.
Right.
So yeah, you need trigger to overcome this, and I think this the ability to concentrate with RNA, for example, provides such an ability. Yeah.
Thank you.
Okay.
Thank you very much.
Gina, you can ask question directly. Bina, you should be able to ask. Yeah, go ahead.
Okay. Okay. Hi, Markus.
Hi.
I have a couple of questions. One is use of RNA in your experiment. Is there a background or a physiological significance to performing that experiment and that observation?
Yeah, sure. I mean, maybe the best motivation for doing is that when you purify amyloid fibrils of tau from the brain, right, it's strongly associated with RNA. So also it doesn't really contain a traditional RNA binding domain. It really likes to bind RNA. So you find RNA, a lot of RNA bound to neurofibrillary tangles. Of course, stress granules have a lot of RNA where tau has been observed to localize. So I think there's a very good support for a role of a tau RNA interaction in a disease process.
My quick second question is this going to be like a generic feature of intrinsically disordered proteins to undergo phase separation and form droplets?
Yeah, so basically that it is, and this is also a fair, let's say, argument for criticism that, I mean, this is a physiochemical process, right? And especially if you think about RNA-induced phase separation, basically you will be able to get any intrinsically disordered protein to phase separate with RNA if you just choose the conditions right, probably. So that's a fair argument. So this refers then back really to your first question that it really is important to see what is the connection really to the disease process in the neuron.
Okay, thank you.
Sumei Zhang, is that right? You should be able to ask a question. Yeah, go ahead.
Yes, thank you. So this is a really very nice talk, a lot of new information. I have a question about tau and stress granule in the cell. In your study, it seems to you first induce the stress granule, then you add recombinant tau to the cell through the membrane somehow with maybe damaged membrane. Is that true? And can you see the colocalization of tau and stress granule with overexpressed tau in cell?
Yeah, thank you for the question. So yeah, this is actually, I mean, this is not an assay we developed. This is actually a very old assay which was originally developed by Dirk Görlich to look at nuclear pore transport, and then was sort of adopted by Dorothy Dorman to FAS and now for tau. So you permalize the membrane digitonian that the protein goes in, right? So it's a artificial assay with no doubt. It has the advantage that you really can provide specifically modified protein, which is otherwise potentially a little bit difficult, right? And that's what we used it for in this specific case. But I think in Ben Wolosin's work, I think I would refer you to, I mean, he has shown in a number of papers that tau is co-localizing to stress granules in diseased conditions. And the modifications of this, let's say, process by modification of TR1 or so affects the disease progress. But this is really Ben Wolosin's work and not ours.
Thank you.
Caesar?
Yeah, thank you, Markus, for this very interesting talk. I was wondering, in your phase separation experiments, you first put, if if I did understand it correctly, you first introduce the post-translational modifications into tau, and then you check on their ability to phase separate.
Mm-hmm.
Have you tried the other way around? Or I don't mean, I mean, is it possible that once the tau phase separate, is it possible for the kinase to have access and introduce post-translational modifications into tau?
We haven't looked at this in detail for this particular project, but on a more general level in terms of phase separation, that is clearly possible, right? So in our work on, for example, the C-terminal domain of RNA polymerase II and phase separation related to transcription, that's exactly the experiment which we did, where we sort of formed the droplets and then added the kinase, and then actually the kinase can go in and modify the droplets, or in this particular case, actually dissolve them. And there's a number of studies in agreement with this. So generally, the kinases actually can go in, and this is makes sense, right? Because really, one key property of phase separated, liquid phase separated states is that molecules very rapidly go in and out, right? It's not a static scenario.
Okay,
thank you very much.
One very last question. Long Cheng, go ahead.
I have a couple more questions. Thank you so much. So, I have one question in terms of your microtubule polymerization as induced by the LLPS. Actually, in this microtubule polymerization, you don't give DTP and also not standard BBRB buffer. And so I know it's very challenging to look at the microtubule dynamics in the same system, but it's durable can look at the stability. So I'm wondering, so is a less stable microtubule or more stable microtubule compared standard polymerized condition, even with the monomer tau? And what's the pathological significant, this microtubule is represent a specific population in vivo, like a more pathological or more physiological, like how stable, or how to understand this microtubule in vivo? Yeah, that's my question. Thank you.
So the microtubule properties and how they differ in different assembly conditions is a very important question. We haven't looked at this, I should say. Of course, there's also I mean, it would also be interesting to look, for example, at the dynamic ends and so on. And there have been suggestions made that tau maybe condensates at the dynamic ends of microtubuli where they grow. Yeah, a lot of interesting questions. In terms of phosphorylation, the 8180 is believed to be strongly related to disease or clearly related to disease progress, because there are these very nice antibodies which are recognizing this epitope at phosphorylation 3 and 231, and one can show that in various model systems that 3 and 231 gets phosphorylated with increasing disease progression. So one hypothesis which you could make based on this data would be that because in the axon there is tubulin polymerization from non-centrosomal sites, and if you then have the tau phosphorylated at threonine 231, our data show that this absolutely blocks this tubulin polymerization. So if this tau condensates in the axon would provide sites for non-centrosomal tubulin polymerization with phosphorylation 3 and 231, this would be really blocked, and we would expect that this have a negative effect on the cell.
Yeah, thank you very much.
Okay, so it's 11:30. So let's thank the speaker, and thanks to all the participants for excellent questions. Thanks, Markus, for your virtual class here.
Okay. Thank you, Rams, for invitation and for organizing. Fantastic.
That was a great talk, and
have a nice day.
Very much. Lots of questions, but great results and great science. Take care.
Yeah. Bye-bye.
Thank you, Markus. It's he. He left.
So next
Yeah.
week, all of you guys, so some of the Europeans missed the talk today because of the time change. Hopefully next week, people will realize that we are changing time tonight in US. So look for EDT. So we are saving 1 hour time. So please join us 10 o'clock